So good morning. Today we will be discussing Mendel's laws of inheritance. So Mendel's laws of inheritance were given by Mendel. He was the first to perform quantitative analysis of inheritance and postulated the laws of inheritance. Some basic terms that we must be clear with. So a phenotype, it is basically an external appearance of the trait or an organism as a whole. For example, you may be looking at morphology, a plant may be tall or dwarf. The genotype. Genotype basically refers to the genetic constitution, the actual genes or the constitution of genes that are responsible for the phenotype. And many genotypes may have the same phenotype. Diploid, as you all know, cells in higher organisms carry 2 copies of the genetic material, and each gene in a sense then is present in 2 copies. Alleles. Alleles or factors are defined as variant forms of the same gene present at the same locus on homologous chromosomes in different organisms. So alleles, as I said, these are variant forms of the same gene, and each of these variant forms is responsible for a contrasting trait. So generally there are 2 alleles, of which one is dominant, that is, a single copy of this allele is enough to give you the phenotype, while the other one is called recessive, which has to be essentially present in 2 copies if the phenotype is to be shown. For example, in pea plant, and we are considering plant height, so capital P represents the dominant allele, that is, capital T, or capital T and small t will give you a phenotype of tallness, and small t and small t together will give you a phenotype of dwarfness. So this is here, so this is homozygous dominant, both alleles of the same type and of the dominant type, so this is called homozygous dominant, this gives you tall. This is the heterozygous individual where one is the dominant allele, the other copy is of the recessive allele, but still the phenotype is tall because one copy of the dominant allele is enough to give you the phenotype. And then you have small t together. This is homozygous recessive, and this results in dwarfness. The model that Mendel chose was Poisson-Sativum, and there are several reasons for it. There were several observable alternative forms for the trait, which we'll see in the next slide. Then the plant is self-pollinated, but it can also be cross-pollinated, which is important for experimentation. And then it has 7 traits which differ very clearly with each other and could be utilized. These are the 7 traits. For our purpose, we'll be using plant height, where we have the trait tall, and this is a contrasting trait dwarf. Likewise, seed color, yellow, contrasting trait green, seed shape, round, contrasting trait wrinkled. Basic experimental procedure. So what Mendel did was first take pure breeding plants of contrasting traits. Pure breeding means that every time you self them, you will get the same trait again. So for example here, you are crossing a pure homozygous tall with a pure homozygous recessive, bringing the 2 traits together and observing raising the first filial generation. So these are the parental generations or P1, and what you get, or the offspring that you get from this cross, are known as the first filial generation or F1. And then he self the F1 plants to raise what is known as the F2 or second filial generation. So this is the typical experimental plan that Mendel followed for all his experiments. So here it is, you have a parental generation, one is pure tall, the other is pure dwarf, and then you cross them together, you get the first filial generation, and what you observe is that there are all tall plants. So no dwarf is observed in the first filial generation. And then again you do a selfing of this, and what you observe in the F2 generation is that the tall and dwarf plants segregate in a ratio of 3:1. Now how do you explain this? So Mendel's findings, first, F1 resembles only one of the parents. For each of the 7 traits examined, Mendel noticed that F1 hybrids always resemble one parent and not the other. The phenotype expressed in the F1 generation was termed dominant, and the other phenotype that was not expressed was known as recessive, was termed recessive. And surprisingly, as mentioned earlier, in the F1 generation, dominant and recessive forms reappear in the ratio of 3:1. So how do we explain this? So first law that Mendel gave was law of dominance, and he says that in the cross of parents that are pure breeding for contrasting traits, only one trait will appear in the next generation. The trait that appears in the first generation or the first parallel generation is known as dominant. The other trait is called recessive. Then he gave a second law based on this monohybrid cross itself, and basically what, as mentioned earlier, it was observed that in F2 generation, dominant and recessive forms reappear in the ratio of 3:1, and this is explained by the law of segregation. So what happens is, so here is a flower plant here, this is a dwarf plant, this is a F1 generation, this is forming only one type of allele, this is a gamete which carries a T allele, then this forms only one type of gamete which carries a small T allele. During fertilization they come together forming the F1 heterozygote, so this is a heterozygous individual with the phenotype of top. And then this is crossed again to get the F2 generation where they segregate into 3:1 ratio. So how does that happen? So this happens like this here. So when the F1 individuals are selved, they form two types of gametes, the capital T gamete, meiosis, the small t gamete, likewise the capital T small t. And then, so alleles segregate during gamete formation via meiosis, and the paired condition is restored by a random fusion of gametes during fertilizations. So these are gametes from the female plant, these are gametes from the male plant, and when they fuse together, they give you in a Punnett square. This is the distribution. So if you see here, homozygous tall, homozygous dwarf, heterozygous tall. So if you look at the phenotype, this is 3:1 phenotype, tall versus dwarf, and if you look at the genotype, this is 1:2:1. Right? So that's it. So then Mendel went on to dihybrid process. Here we are taking two characters together and studying their inheritance. And what we want to see is whether when you take two characters together, do they influence each other or not. So here what we have taken is yellow round, what Mendel proposed yellow round, this is homozygous dominant, versus green wrinkle, this is homozygous recessive. You have one type of gamete here which carries the dominant alleles for yellow and the shape, and you have the other one type of gamete here which carries the recessive alleles for color and shape. So basically green and red. And then you freeze your F1, the two come together, giving you heterozygous yellow round. Right? And then these are selved again. When this meiosis takes place, there are four possible gametes that form. So you have one segregation where capital Y and capital R come together, you have another segregation, capital Y small r coming together, small y capital R coming together, and small y small r coming together. So this is the gametes that are formed, and then they are selved. So when you self them and put them into a Punnett square, this is the distribution that you get. Very clear, if you look at the phenotypic ratio, you have 9 yellow round. Right? Then you have one green wrinkle, and you have two new combinations. This is basically your yellow wrinkle and green round. So yellow wrinkle is here, here and here, and green round is here, here and here. This is green round. Okay. So what does it mean? What does it mean in the context? So if you look at the law of infinite assortment, it basically states that when you're considering the two genes together, inheritance of two genes together, they are actually not affected by each other. For example, here in this case, if you look at the individual trait, the seed color, yellow is to green. If you look at this, so this is 12 here versus 4 here. 12 is to 4, or actually it is 3:1 when we simplify this, which is typical monohybrid Mendelian cross ratio. And if you look at the phenotype again, homozygous dominant versus heterozygous versus homozygous recessive, the ratio is 4:8:4 or 1:2:1. So these four are homozygous dominant, these are heterozygous, and these are homozygous recessive. Here. Likewise for the other trait, if you look at round is to wrinkle, this again gives the 3:1 ratio, and again this simplifies into 1:2:1 ratio in terms of genotype. The two genes assort independently of each other and follow the same ratios as in a monohybrid cross. That is, they do not really affect each other in any way. So if you consider the inheritance of two or more genes at a time, their distribution in the gamete and in the progeny of subsequent generations is independent of each other.